So in that what happens, hemolysis is happening, and due to that it will also cause jaundice in the newborn, that baby. Okay, so what is happening, what the body is going to do, that the infant baby body is going to do, so it is going to compensate of hemolysis, so by doing of rapid production of RBC, rapid production of RBC. So where the production of RBC taking place? Can someone tell me? Bone marrow. So as we know that the individual, here the stage is for infant, the newborn baby. So during that time their spleen and liver is also taking role for making production of RBC. Okay. So many immature, we can say, immature of erythroblast stages can be released directly into the circulation, and that condition, because of the immature of erythroblast cells are released directly into the circulation, that's why this disease has been called as erythroblastosis fetalis. So we have seen the diagnosis we discussed, and we found out at the genetic level how it is happening, and we saw that all the hypotheses also, right students? Everyone? Till here everyone understood? Can I just get the voice for listening? Thank you. Now please, we'll come back to our next topic today. We'll discuss polygenic inheritance. So first we need to start from that. The Mendel has chosen different colors. Okay, different color for the like he has started the work with the plant. So he talked about color and size, yes, length. So all this phenotype, he has done experiment that he has chosen two different individuals from the populations. So we know that there are the various type of characters are present in the population. Yes or not? So yeah, this kind of various kind of characters can be found in the population. It is become quantitative characters. It has become quantitative characters. So this quantitative character is only going to indicate the polygenic inheritance. Okay. So in this, what is happening? Poly means, everyone understand poly word is used where? Many, right? So we can just make the point here. Cumulative effects of two or more genes. Phenotypic characters. So this is the definition of polygenic character. Here the cumulative effects can be happen from two or more genes for the single phenotypic character. Okay. And we also have learned when it is just a converse of polygenic, when we are talking about, we already knew that single gene can also do many different type of phenotyping. Yes. So it is a converse of polygenic. In the polygenic, what happening? There are two or more genes are involving for the single phenotypic character. As we already know that single gene can also affect to many several phenotypic characters. That is the converse of pleiotropy, right? So these examples can be used for the converse of So understood, students, till here? Can I get the voice? Skin color in man. We will discuss is an example of polygenic inheritance. So you know that skin color is same in all the human beings or various? Okay. It is because of the polygenic of genes. So this example can be considered as quantitative characters. Because of that only the skin color is going to become different. So the skin color it can be controlled by 3 genes. It should be mentioned, controlled by 3 genes. So that 3 genes which is inherited. Inherited means they have come from parents, exactly. So let's it has been given the example of 3 genes should be A, B and C. So 3 genes name given. Let's make too easy this A, B, C. Understood till here, students, everyone? Okay. So that was a dominant capital letter we have used for the dominant, and you can just make like this: dominant A, B and C. We'll use recessive, will be use A, B and C. So because they are number of genes, so the plural of this, so this is singular, right? So when it is genes, so we can also use the word alleles because they are the group of genes, right? So they are for the dark skin, and the small recessive alleles which we use small letter, it is for the light skin. Okay, so whenever that the genotypes will be like this, you know that they will be in the pair. If you remember Fisher hypothesis, yes. Yeah, exactly. Their genes are expressing for the pigmentation, that's why. So it is becoming dominant over there in their population. So it is a diversity of genetics. So it is opposite for the, if we see that, of course, so if recessive is completely there, so no dominancy, so there the all people will be albinism. Albinism is the again it is called as disease, yes. Yeah, but that condition is different. We cannot make them very light people, disease people. Exactly. Melanin is a pigment that is absent. Then we will see that what is happened after mating and doing reproduction. So their genetic material is mixed. What happens? So that one more result will become is called as intermediate color. Yes, intermediate. Yeah, we can see here they will become combination. So this is showing intermediate shade. Okay. So we can see that when this and let make cross with this. So what will happen? This will make genetic contributions, right, to the skin darkness. So this again that the variety of shades can be observed when these gametes are meeting. So when the skin color we are getting because of we are getting this combination from where? Parents. So they will be called as these gametes looking like heterozygous, right? Heterozygous organisms. So we know that the for this color darkness you talk about or light skin, for everything this that the alleles is main reasons that the group of genes are responsible for that it is going to work on that, it is going to determine that it should be a dark color in the person or it should be the light color in the person. Okay. And also there can be one more reason that which is a common reason. If someone is exposed to sunlight directly, that can become also the skin color will become dark. So that condition cannot be considered for genetics. That is because of environmental factor, right? So the cells of that skin is going to little damage, and because of that the color will be having shades of color can be seen because of the sunlight. Everyone understood till here? Yeah, bomb blast, I think that will not leave the face of person, I think so. Yes. How it will leave? It will damage the whole. What do you say? Yeah. Radiation, you can say radiation can also affect. So there are the many other factors can be discussed that will be come under into the research point of view. Okay. So until we do not have evidence, that will be just like a hypothesis research. Okay, student. So understood till here, everyone? Yes. Now we will talk another topic that is called as a sex determination. So everyone know that is a identifying that which gender it is, male and female. So Alhamdulillah, I will really appreciate to the government that they have just allowed this kind of method that sex determination is banned, because from the ends the long time in the ancient times, since peoples are killing to the girls, okay, girl child. Are you getting? So they are escaping from that. They don't want to bear the girl child because they feel disrespect that someone have girl child in the family. Even Allah subhanahu wa ta'ala has really warned this thing in the Quran, if you have heard. Yes. So to stop this kind of killing the girl child, of course the government has taken decision that is sex determination should be banned. So that's why I appreciated. You understood why I appreciated. Yes. Otherwise there are a lot of things which should not be appreciated. Okay, fine. Please come back. So sex determination can be done. First of all, there are the many methods. Of course, the organs is identified in the embryonic level or at the genetic level and chromosomal level, it is identified that they have which type of chromosome present in this organism. So that time also sex can be determined. Yes, sonography at the stage, actually what happens is scanning. That process is observing the whole abdominal inside, the movement and the structure of the baby. So that can be determined by the doctor, but it is clearly strict to them that they should not inform to the patient. Are you getting me? But they do not say. I do not know that. That is their personal thing. Sometimes they inform or they do not inform. I do not know. But the most of people having, we can say that most of people, because this is the word of Allah subhanahu wa ta'ala, most of people, they have darkness on their face when they just listen the news of that they got the girl child, because Allah knows best. Yes, He knows. And those, suppose for an example, we knew this thing. What is Allah wanting from us? Allah is saying that you all people are wrong. If you listen my word, that means you accept that you will not do that thing which I just curse on that thing. Okay, understood, students, everyone? So we are talking about sex determination and its reality. So let's talk about its sex chromosome first in this. So which are the sex chromosomes in male? Okay, and in female? So one pair of sex chromosome, yes, remaining is autosomal chromosomes. So this sex chromosome can also be called as allosomes. Okay, and we also know that the other chromosomes called as autosomes, which is 22 pair, and this is one pair. So 22 is becoming 44, one pair is becoming 2, total is becoming 46. In every single human being cells is going to carry in its nucleus 46 chromosome. Understood, students, everyone? Yes, of course. If someone asks that, is there any difference between autosomes and sex chromosome? Absolutely, there is difference. Okay, it is distinct from other chromosomes. Sex chromosome is distinct from other chromosomes. So that experiment which is going to prove that how the sex chromosome is distinct from other chromosome, that has been held by King, the scientist. Okay, so we have done this. Now let's move to the next slide. So what he was doing in his experiment, let's talk about. He was observing nuclear structure. What he was observing? Yeah, so of course you know that nuclear, this word has come from where? And nucleus is, that means he's focusing on cell. Yes, so nucleus of the cell he is observing its structure, and through where, in which stage? Just taking the tissue and cutting it, putting it under microscope? No. So he's seeing this nuclear structure from the stage mentioned, that is spermatogenesis, because we learn in the human reproduction how the spermatocyte is formed: primary, secondary, ultimately spermatozoa. All students remember? Yes, sorry? Yeah, it is very beginning, we can say. Spermatogenesis means, of course, at that time it is happening until it is become sperm. They will be having two set of chromosome. Yes or not? Yeah, that's why. So we should remember spermatogenesis is including two set of chromosome stage also, and ultimately in the terminal it will become one set of chromosome, right? Haploid. Yeah. So what he has observed during that time, so he found it that 50% of spermatozoa. So when you are talking about spermatozoa, how many chromosomes should remain over there? No, I mean set 50%. Yeah, 50% of spermatozoa. No, we are talking about spermatozoa, but I am saying spermatozoa have chromosome set 2n or this single n. Yeah, exactly. Yeah, that we need to very clear about it. So what he observed that 50% of spermatozoa receive their structure like a sperm, okay, and 50% did not receive. He observed that 50% of spermatozoa received this structure, okay, and 50% they did not receive through the spermatogenesis process, okay. Okay, so he gave that structure name, suppose it is X body, okay. So the structure name has got X. But he could not able to explain the significance of this structure. That the drawback of this scientist. So ultimately then this structure was investigated by many other scientists. So the drawback, no significance discussed, okay. So you can see that at the scientist level also we can just have an example that how many things are still for every human being is like a research and learning. Yes, just some of them that they might be little ahead than you, just like that. Yeah, see other scientists, they just took his experiment to proceed that what he had done. So let's take it as a what to do with that. Okay, so what the other for the investigation of scientists, what it has brought, that the we can see that conclusion was he was talking about the chromosomes, okay. He was talking about the chromosome at the spermatogenesis level. So that's why that chromosome name has got X chromosome, as we already know. X chromosomes, when they have studied at the nuclear level, so of course they got then later the chromosomes mapping also, right. So the scientist name mentioned here that some more Stephen and Wilson. So they identify first Y chromosome. They identify first Y chromosomes, okay. So it has been now helpful for sex determination, because they totally in the whole of chromosome mapping they found it that which is X and which is Y. Everyone is understanding? Yes. So they first determine into the example that in the lower organism, so that has been named mealworm, okay. Mealworm they have observed it, and one more example given, genus Tenebrio Molitor. Understood till here, students? A very good question. Well, see, the all part of learning is important when we want to become perfectly in the study. So, but of course, as the point of view that attending questions and this thing, okay, so this will definitely become some basics in some of the question to you. So if you understand all this sex determination part, so it will be helpful to you. Are you getting what I'm trying to say? So any dot is missed, you know, so you cannot, you will be able to find where is that dot. So you will not, if you won't find that, so then how you will proceed it. So it's better that all dots must be connected. But whichever that, if something is not required, I'll definitely help you here that to not take that part. Okay. Welcome. So ultimately sex determination has been done with that Y chromosome is present or absent. On that basis sex determination can be done, that is male or female. Yes. And one more thing we can just memorize, that most important, that is they are homogametic, those who have the same type of chromosomes, and those who have a combination they are called as heterogametic. Okay, so this is the word which we should memorize it. Why it is heterogametic? Because it is and there because of that they will be made dissimilar, and they are similar like chromosomes. That's all. So this also we should memorize it. So ultimately this sex determination will be based on, first of all, the chromosomal theory. Okay, you need to know that each number of chromosome, how they look like. Then need to discuss genic balance theory, how the genes are going to express and they're doing it in the sex determination. And haploidy method. So what is that? Haplodiploidy method, that we are going to discuss that in which stage the chromosome is becoming haploid and which stage it is going to become diploid, right? So these three methods should be carried in every single sex determination if the perfection, if it is required. Otherwise I have told there are many methods to identify the sex as a gender, that which is male and female. So today there are many processes are also available to identify at the genetic level also, at the gender level also, and even the scanning process can also able to recognize by the expert. All the students getting me here till sex determination. So like this sex determination part is completed. So the some more part we will discuss in this, that is called as heterogametic sex determination. Let's talk about that. Today there is one more class, I think so. Homogametics, there is no further discussion has happened. So that's why the heterogametic will be discussed in all this right now. Okay. Let me take the attendance please. So everyone understood till here sex determination, today our topic. Okay, girls. Okay, fine. Time just will do continue even in the next lecture inshallah. Heterogametic sex determination. So by the name of heterogametic we found that they are not similar chromosome. Yes. So sex determination is done through the chromosomes. So that condition making them the name should be given determination process. So allosomes you say or chromosomes you say, everyone is understanding? Allosomes or chromosomes. So when it is happened, that condition, that terminology will be called as genetic or, so without chromosome there is no genetics, right? Because chromosome is going to become both for all the genes and DNA. Understood these points till here? Yeah, tell me. Yeah, but here, yeah. Without let me tell you, in the beginning only I have told you what is the allosomes, if you remember, right? Allosomes, I told that was a no. Allosomes is just used, the word is used for the sex chromosome. Okay, please remember this, right? Understood now? Okay, please remember that. That's why some point when dot is missed, no, so it's fine that you ask me, and I hope that now you understood. The allosomes is name given alternative for the sex chromosome. Okay. So when the sex determination is the sex determination process is happen through the chromosome, sex chromosome, or it can be also called as allosomes. That terminology will be called as genetic or chromosomal sex determination. Okay. After that, I told that of course the sex determination cannot be happen without the chromosome. Okay, because chromosome is becoming the main medium where all the genes will be present and all the DNA will be present inside of the chromosome. In the beginning only I told about that. So clear, students, everyone? Okay, so they are dissimilar gametes. We can say they are dissimilar gametes. Okay. Okay, the main now the time of determination of sex, when it can happen? Fertilization. Okay. So that condition has got one more name. You should remember it by syngamy. That is fertilization. Yes. So it will depending on the two dissimilar gametes. One is coming from male and another is coming from female. Okay. So we know that when the both gamete is mixing, that condition is making heterogamete. But when they are at the individual level, haploid level, they were homogametic. Yes, homogametic parent. So we can just mention point, two homogametic parent gave the heterogametes, right? So now the another topic which we should discuss, that will be male heterogamete, right? So because they are dissimilar chromosomes. As we have just 2 minutes more left, I think so. Human are correct. All the females are homogametic. Yeah, but the one condition, after slowly when we will move, we will have one more diversity that we will learn there, that female also have now got heterogamete, but that is in different organisms, that we will discuss. Okay, fine. Birds. So in birds you can find the heterogamete in female also, but as per now in the human beings it is homogametic. So this part we will be able to cover because it will be disconnected within I think 1 minute. So let's try how much we can do. I think I need to save this. Fine. So everyone understood heterogamete word is used for the male, okay? Just make it here. And homogametic word will be used for female. So we can find out that heterogamety can be of two types. What are those? Right? So male sorry? Are you understanding this? Male heterogamete, there must be the two kinds, XX and XO type, right? So, and XX, XY type. So this we can see that from which that XS is referred to the female, and this type will be referred to the male. Understood till here? Yeah, XO, that remaining part I'll explain you in the next class. So till that time, because it will be disconnected, so again we'll need to start from beginning. So it's better we'll discuss in the next class.